Hello, and thank you to the committee for selecting this project for this presentation. My name is Elysia Saputra. I'm a PhD student in the computational biology program jointly offered by Carnegie Mellon University and the University of Pittsburgh. And today I'll be talking about a comparative genomics method that we're currently developing, which we call phyloConverge, a likelihood-based approach to identify genomic regions underlying phenotypic convergence at nucleotide resolution. So I'd like to start this presentation by pointing out the amazing phenotypic diversity observed across species on Earth, and to highlight that understanding the molecular basis of this diversity is one of the primary interests in the evolution community. And to understand this phenomena, there's been a lot of investigations that's been done to study both the coding and non-coding portions of the species genomes in order to find the nucleotide level changes that underlie the organism level morphological changes that we can observe. And to get to this end, one of the strategies that we can use is to sequence the genome of species with extreme phenotypes. But this strategy also has its own set of challenges because species can differ from their nearest relatives in millions of base pairs and in multiple traits. And so this would complicate our ability to infer associations between genetic elements and the trait of interest. And to overcome this challenge, convergent evolution is a really useful tool that we can use to make these type of associations. Convergent evolution is a process in which multiple lineages independently develop similar characteristics. And here on this slide is an example of such a convergent event in which several independent lineages have over time developed the ability of echolocation. And these include several species of bats and several species of toothed whales, such as the sperm whale and the bottlenose dolphin, all of which have over time acquired the ability to emit biosonars to make sense of their environment. And so when such a convergent event occurs, this can allow us to map genotype to phenotype because presumably the convergent phenotypic changes that we observe across these independent lineages would have resulted from convergent molecular changes that occurred in response to stimulus Russian pressures. And with this assumption, there's been a lot of studies that's been done to find convergent substitutions in the level of amino acids, and at a higher level to find convergent substitutions of nucleotides. And for the most part, these approaches have been really useful, but they also often suffer from low statistical power. And the reason for this is because the assumption behind these approaches is that the morphological change that we observe result from a specific identical mutation occurring across species. But this may not address all the possible molecular drivers of the morphological change because complex phenotypes typically arise from a complex multigenic process. And so this idea then motivates an alternative approach at even a higher level, which is to find convergent evolutionary rate shifts at the level of genetic elements. And the idea behind this is that genetic elements that are important for survival or development would be expected to evolve slower compared to the neutral rate. And an example of the application of this idea is in the realization of constraints, in which genetic elements that are no longer important for survival or development would be subjected to a relaxed selection pressure. And to illustrate this scenario, we'll take the example of the subterranean mammals, which include several species of mole rats and moles, shown as the red branches in the evolutionary tree that we see in this slide. And one of the key traits shared by these mammals is that they have diverged visual structures. And so when we look at the evolutionary tree of a specific protein that's involved in the development of the lens, shown on the right here, which is the limb-2 protein, we can see that relative to the proteome-wide average tree, the evolutionary rate of this protein across the mole and mole rat species is faster compared to the rest of the mammals in the evolutionary tree. And so this means that there's a correlation between the acceleration of evolutionary rate of this protein with the evolution of the subterranean mammal phenotype. And this is an example of how evolutionary rate-based methods can be used to associate genotype to phenotype. And so this type of evolutionary rate-based methods have then become more and more popular because they have been successful for associating genotype to convergent phenotype. And this is particularly the case for studying coding regions, but when we work with non-coding elements, there's a different set of challenges that we have to address. So firstly, all of the existing methods that are currently available require the users to supply predefined coordinates of the genetic elements of interest. But the notion of a genetic element is often not well understood for non-coding elements, and therefore is difficult to define. And secondly, the evolutionary process of regulatory elements tend to be variable. And so there is a need for a more rigorous accounting for this evolutionary process when we want to detect shifts in evolutionary rates. And lastly, the distribution of information content across different portions of an enhancer may be non-uniform, especially for pleiotropic enhancers that are involved in regulating multiple functions. And so given these challenges, the aim of the study is therefore to design a computational strategy for associating genotype with phenotype that rigorously estimates evolutionary rates of a genetic element and enables high-resolution scoring of divergence without requiring prior definition of genetic elements. And because of that, we then develop a method called phyloConverge, which is a likelihood-based phylogenetic method for finding convergent nucleotide association rate shifts. The method takes as input three things. First, it takes a phenotype tree. Over here, the red branches signifies the foreground species, or the species with convergent phenotype. It also takes in the multiple sequence alignment of all the species in the phylogeny. And finally, it also takes in the neutral tree model that represents the neutral rate of evolution. And given these three inputs, phyloConverge makes use of functionality that's been implemented in the phyloP function in RFAST, which uses maximum likelihood to estimate two branch scaling factors. The first scaling factor is a global scaling factor that determines if there's a phylogeny-wide acceleration or deceleration of evolutionary rate observed for a particular element. And the second scaling factor is foreground specific, and it determines if there's a convergent shift that's observed only among the foregrounds relative to all the other species in the phylogeny. In addition to that, phyloConverge also uses a phylogeny-aware tree permutation method that we previously developed called phylogenetic permutation. And what this method does is it produces a set of null or fake phenotypes that allows us to statistically calibrate the output of the base phyloP method. And the idea behind that is that the assumptions made by these methods may not always be generalizable to all cases, and therefore we'll have to empirically correct for that. And in short, permutation takes in the observed phenotype values, uses the values to do phylogenetic simulation, and produce a set of simulated branch lengths from which we can select a new set of foreground species that will form a null or fake phenotype. And the selection is performed such that the number of species that we select matches the number of foregrounds that we find the observed phenotype, and such that the relationship between the new foregrounds also matches the relationship in the observed foreground. And after we produce, for example, 1,000 of these null phenotypes, we then subject them to the same phyloP scoring, such that at the end of the day, we'll have acceleration scores calculated for both the true phenotype and the empirical null phenotypes, which finally will allow us to calculate acceleration scores that are bias corrected and can be performed at any resolution. So we then benchmark our method using a data set that was previously analyzed by Rosito and colleagues, which is on convergent evolution of the subtree mammal phenotype. And this data set contains a multiple alignment of 24 species with 4 mole rat and mole lineages that can serve as our foregrounds. And from this multiple alignment, Rosito had also calculated 500,000 cis non-coding elements using FastCons, which will be the base of our initial analysis. And what is notable among these 4 lineages is that one of the common traits they share is eye degeneration. And in fact, they're practically blind. And so we would expect that eye-related regions will experience relaxation of constraint and therefore will have acceleration of evolutionary rate. And to see if that's the case, we first use phyloConverge to calculate acceleration scores for the 500,000 CNEs, and from that identify the top-ranked quote-unquote mole-diverged CNEs. And we then correlated the mole-diverged CNEs with open chromatin regions across different tissues. For this, we use several sets of annotations of mouse embryonic ataxic data. And we found the correlation using by finding the number of intersection of the mole-diverged CNEs with the open chromatin regions in each of the tissue-specific annotations. And when we do that on the eye-specific annotations, we see that there's an enrichment of mold-diverged CNEs in the open chromatin regions of eye-specific tissues relative to the null, which is 1,000 sets of randomly chosen CNEs from the total set. And we also note that the mold-diverged CNEs identified by phyloConverge shows a stronger signal compared to a competitive method called forward genomics, which was previously used by Rosito and colleagues. And we also tested a bunch of control tissues for which we don't expect to see enrichment. And in fact, that is the case that we see over here. And so based on this, we can conclude that phyloConverge seems to be working pretty well. And after we identified our mold-diverged CNEs, we then did some functional enrichment analysis to see what functions the CNEs may play a role in. And for that, we use a tool that's called GREAT. And the results showed that there are 49 human phenotype annotations that are enriched with mold-diverged CNEs. And plotted here are the top 20 of these 49. And we note that these top 20 phenotypes are relevant to the subterranean phenotype. And notably, a lot of them are involved in ocular and neuronal related functions. We also performed this analysis on the canonical pathways and identified that there are 25 total pathways that are enriched with mold-diverged CNEs. And the top 10 that's illustrated over here are also highly related to ocular and neuronal relevant processes. And consistent to what we observed before, together, this suggests that the eye degeneration that occurs due to subterranean adaptation may also be accompanied by some rewiring in neuronal processes. And next, we also demonstrated the ability of phyloConverge to perform acceleration scoring at higher resolution. And to do this, we take our top-ranking mold-diverged CNEs, and we scored every 10 base pair position of the CNE for this acceleration, as shown by this illustration over here. And after we do that, we correlate these scores with the coordinates of about 800 transcription factor binding site motifs genome-wide, and this allows us to finally calculate the aggregate score for each of the TFBS motif. And after we do this, we find out that there are 179 TFBS motifs that are significantly diverged genome-wide, the top 10 of which are shown over here. And similarly to what we saw before, there are a lot of these TFBS motifs that are known to be involved in ocular and neuronal development. So this, again, is a consistent finding. And what we would finally want to be able to achieve is to perform the scoring at even higher resolution and see if this can allow us to identify accelerated regions at really high resolution, for example, at the TFBS level, without requiring users to supply predefined regions. And here, while we have some preliminary results from that, and the illustration that's shown here is on a known enhancer of PAX6, which is master modulator of eye development. And we can see that within this enhancer region shown in blue, there's a specific portion of the enhancer that is actually significantly accelerated and correspond to known transcription factor binding sites that are known to regulate this particular enhancer. So this demonstrates the potential of the method to be able to perform this analysis at even higher resolution. And so to conclude, we have developed a method that is based on a maximum likelihood framework for associating genetic elements to convergent phenotype. And our method, phyloConverge, can also perform high-resolution scoring in a computationally tractable manner. We also showed that using a well-characterized phenotype, our method was able to improve on the performance of competing methods and produce biological signals that are relevant to what we expect. And our preliminary results also showed the potential ability of our method to perform high-resolution segmentation of CNEs into functionally relevant subregions. This concludes my presentation for today, and I'd like to use this opportunity to thank both my advisors, Dr. Maria Jacquina and Dr. Nathan Clark, as well as all of my lab mates, all of whom have helped me in one way or another in the process of this work. Thank you again to ISMB and the selection committee for giving me this opportunity to present, and I'll be happy to address any questions. Thanks.